Good day, ma'am. I'm Maria Cristina Ramos together with my groupmates who is here to explain or to elaborate to you what protein in biology is, together with its type, functions, also characteristics, and some examples. So now, before we move on to its characteristics, let us now define what protein is. So proteins are fundamental micromolecules in biology playing diverse and essential roles in the structure and function of living organisms. It is composed of long chains of amino acids, proteins, and intricately involved in process ranging from cellular structure and support to enzymatic catalysis, signaling, and immune responses. The building blocks of proteins are amino acids. The unique sequence of these amino acids in protein structure is determined by the genetic code encoded in an organism's DNA. This sequence often Proteins contribute significantly to the structural integrity of cells and tissues. Fibrous proteins like collagen provide strength and elasticity to connective tissues, while globular proteins such as hemoglobin facilitate oxygen transport in blood. Enzymes, as a specialized class of proteins, act as biological catalysts, accelerating chemical reactions necessary for cellular process. Moreover, proteins are vital in cell signaling pathways, transmitting information within and between cells. Receptor proteins on cell surfaces, for instance, play a crucial role in responding to external activities. Antibodies, another type of protein, are essential components of the immune system, recognizing and neutralizing foreign invaders like bacteria and viruses. So in summary, or to sum up all of the ideas that I have said, proteins are versatile and indispensable molecules in biology, contributing to the structural framework and enzymatic reaction signaling processes and defense mechanisms that sustain life. Now, moving on to its characteristics or the functions types and some examples of it. Now to continue the discussion, we will now move to the classifications of proteins. So the first one on the list is structural proteins. So what is structural proteins, or what does it mean? So structural proteins serve as the architectural backbone of living organisms, providing essential support and maintaining the integrity of cells and tissues. These proteins are characterized by their role in changing and reinforcing various anatomical structures. One well-known structural protein is collagen, which forms a strong fibrous matrix in connective tissues like skin, tendons, and bones. This collagen network impacts resilience and elasticity to tissues, ensuring that they can withstand mechanical stress and maintain their structural integrity. Actin and tubulin, sorry for my pronunciation, integral components of the cytoskeleton are also vital structural proteins. Actin filaments contribute to cell shape and facilitate cellular movements, while tubulin forms microtubules that provide structural support and serve as tracks for intracellular transport. Beyond their role in maintaining physical structure, structural proteins are involved in dynamic processes such as cell division, migration, and intracellular organization. By contributing to the overall architecture of cells and tissues, these proteins play a fundamental role in the form and function of living organisms. Now moving on to the next one.
So, there are 4 structural proteins, and today, me and Miss Gabby will going to explain how the 4 of them works. But let me start with the primary first, and then secondary, then Miss Gabby will explain the last 2. The primary structure refers to the linear sequence of amino acids that make up the protein chain. This sequence is determined by the genes encoding the protein. The primary structure is a crucial level of protein organization and plays a fundamental role in the overall structure and function of the protein. Proteins are composed of a specific sequence of amino acids linked together by peptide bonds. The order of these amino acids is determined by the genetic code within the DNA. The genetic information in the DNA is transcribed into the messenger RNA or mRNA, which carries the code for assembling the amino acids in a specific order. The mRNA is then translated by cellular machinery, such as ribosomes, into a sequence of amino acids. Each 3 nucleotide sequence on the mRNA or codon codes for specific amino acids, determining the protein's primary structure. The amino acids are joined by peptide bonds, forming a polypeptide chain. The specific sequence of amino acids along the chain constitutes the primary structure. The primary structure influences the protein's folding into its secondary, tertiary, and quaternary structures. The arrangement of amino acids determines the protein's unique three-dimensional shape, which is critical for its specific function. Secondary structure in proteins refers to the local folding patterns within a polypeptide chain. The two main types of secondary structure are alpha helices and beta sheets. Here's how secondary structure works. After the amino acids are linked together in the primary structure, interactions between nearby amino acids cause the polypeptide chain to fold locally. In an alpha helix, the polypeptide chain curls into a helical shape stabilized by hydrogen bonds between the carbonyl group of one amino acid and the amino group of an amino acid 4 positions down the chain. In the beta sheet, the polypeptide chain folds into a sheet-like structure with segments of the chain running alongside each other. This is stabilized by hydrogen bonds between adjacent strands. Hydrogen bonds play a crucial role in stabilizing secondary structures. In alpha helices and beta sheets, hydrogen bonds form between the backbone atoms of amino acids, contributing to the stability of these structures. Secondary structures are building blocks for the tertiary structure of a protein. Multiple alpha helices and beta sheets, along with turns and loops, interact to create the overall three-dimensional shape of the protein. The secondary structure is essential for a protein's function. For example, alpha helices are commonly found in membrane proteins, while beta sheets can form the core of a certain globular proteins.
The third type of structure is known as tertiary structure. It has more folding that occurs in the 3D shape of a functional protein, and this is because of the R groups, or also known as side chains. So, the different R group interactions like H bonds, the static forces, disulfide linkages, and van der Waals forces can influence the folding of the tertiary structure. So the fourth structure is the quaternary structure, and it consists of more than one polypeptide chains. So, proteins can be made out of one or more polypeptide chains, and you can see it in this type of structure. So each of these polypeptide chains can be a subunit, and interactions between them, such as hydrogen bonds or disulfide bonds, can keep them together.
Contractile proteins are primarily involved in the contraction and movement of muscles. They play a crucial role in the ability of muscles to generate force and produce movement. Storage proteins, as the name suggests, are proteins that serve as a reservoir for amino acids or other essential nutrients. They store these molecules until the organism needs them for various biological processes.
Defense protein, also referred to as immunoglobulins, are the central most important part of our entire immune system. These proteins includes antibodies, a blood protein produced by our immune system in response to and counteracting a specific antigen which protects you from unwanted substances from entering your body. It also promotes protection against our foreign bodies. Transport proteins. Transport protein serves the function of moving other materials within an organism. These includes hemoglobin, which is the protein contained in red blood cells that is responsible for delivering oxygens to the tissues, and iron-containing protein in blood that transports oxygen from the lungs to the other parts of the body. Signal proteins. Signal proteins regulates how cells react to different environmental cues. Aside from that, these proteins serve different biological functions, including cell survival, proliferation, polarity, and migration, as well as fate specification and stem cell self-renewal. This also provides the coordination required for the functionality of multicellular organisms and hormones which coordinate body activities.
Digestive enzymes play a key role in breaking down the food you eat. These proteins speed up chemical reaction that turns nutrients into substances that your digestive tract can absorb. Enzymes are essential for digestion, liver function, and much more. Your saliva has digestive enzymes in it. Some of your organs, including your pancreas, gallbladder, liver, also release them. One of the most important roles of enzymes is to aid in digestion. For example, there are enzymes in our saliva, pancreas, intestine, and stomach. They break down fats, proteins, and carbohydrates. Enzymes use these nutrients for growth and cell repair.